We're going to be kind of introducing the field of biological psychology in this lecture, as well as discussing the ways in which context contributes to our behavior. So for those of you who haven't taken a course in biological psychology, it is really just a combination of biology, psychology, and neuroscience. The desire of this field is to understand the relationship between brain and behavior. And sometimes it's actually known as behavioral neuroscience. You can have specializations within this field. For example, biological psychologists who are trying to understand cognitive functions such as memory may be considered cognitive psychologists. Those who are interested in using physiological functions during or measuring physiological functions during psychological testing could be considered psychophysiologists. And lastly, for those biological psychologists who are interested in understanding the effects of drugs on brain and behavior may be considered psychopharmacologists. There are many other subfields or specializations within biological psychology, but these are some common ones that you might encounter. To me, biological psychology is probably the most fascinating and awe-inspiring topic in all of psychology, but then again, I'm also slightly biased. So your textbook uses context in terms of trying to understand behavior, particularly how the brain controls specific behavior or responds to the environment. So context essentially are circumstances surrounding an event, and our behavior is not occurring in isolation. Rather, it's an interaction between the brain's perception of the environment, the behavioral response of the individual. And so context is really what determines the brain's response. So for example, if you go skydiving and you voluntarily jump out of a plane knowing that you have a parachute that's going to save you, your brain and behavior are going to respond very differently, for example, than if you're clinging for your life after being pushed out of a plane. So the context of a situation is going to be very important in determining the ways in which the brain responds to its environment. And we'll be discussing the context of various psychological processes to determine the brain's and body's response. Many factors can influence your daily activities as you attempt to watch this lecture. For example, you might have the TV on or listening to music, and so that might influence your ability to pay attention to what I'm saying. Likewise, if you're hungry or haven't eaten, your ability to focus and comprehend the content of this lecture may differ. So nothing really exists in a vacuum. Some of you might recognize this individual here on the right. His name is Charles Darwin. He's the father of evolutionary biology and essentially developed the theory of natural selection in his writing that he published in the late 1800s, titled On the Origin of Species. What he theorized is that species are uniquely adapted to their environments. Those that are better suited for the environments or can easily adapt are more likely to survive and therefore have offspring, and those offspring will have the traits that made them more adaptive too. And so what Darwin did is he used finches, Darwin's finches on the right, that he kind of studied in the Galapagos Islands, and he noted that they likely evolved from a common ancestor. But what's interesting is that these finches have different sized beaks based on their primary food source. So the finches that largely feed on insects, let me see if I have the pen up, yep. The finches that largely feed on insects have much longer, narrower beaks, for example, than finches that might be feeding on things like nuts that require much bigger, stronger beaks to break through them. So Darwin emphasized the context in the development of species survival, that these species needed to adapt to their food source, which is the context, in order to be able to get the food to survive. And so it was naturalist Ernst Mayr who identified two different contextual variables based on Darwin's theory of natural selection. He said that there were ultimate causes that dictated behavior as well as proximal causes. So ultimate causes of behavior are the long-term evolutionary causes of physical or behavioral characteristics. So they represent the why particular things are the way they are. Proximal causes of behavior are much more short-term and include things like hormone secretion or responses to the environment. So throughout this course, we'll be evaluating the proximal causes of various behaviors as well as their potential evolutionary history when it's relevant. For example, your desire to hang out with friends are the result of developmental patterns of social responses from childhood through adulthood, right? They were kind of determined by this behavior that you engaged in, as well as the neurochemical responses that are associated with social behavior. So particular hormones get released when you engage in social behavior or connect with others. But they could also be the result of ultimate causes, such as greater survival in primates that engaged in social behaviors and worked together to help each other out. So these social behavior, your desire to hang out with friends, has both proximal causes as well as evolutionary causes. So in the 1960s, a research team at UC Berkeley evaluated the impact of complex environments on rat development. You'll notice that we spend a lot of time looking at research that occurred in rodents because rodents tend to be a model organism, and we'll touch on that in lecture 2. But in the 1960s, they were curious to see how environment influenced rat development, specifically rat brain development. So they were interested in understanding the influence of complex enriched environments, which consisted of social groups of 3 rats per cage. Additionally, the enriched environment contained objects that were placed in each cage and replaced several days to ensure that the environment was dynamic, as it would be in the wild if these rats were not living in cages. And then after they were grown up, these enriched rats were compared to animals that were housed individually in cages with no objects, what they call the impoverished environment. After about a month, they took the brains from these animals, removed them, and compared them. And what they found was that enriched rats had thicker cortical areas of the brain known to be involved in higher cognitive functions, as well as more connections among the cells themselves. So when they looked at individual rat brain cells and compared them in the enriched environment to a rat brain cell in an impoverished environment, you'll notice that there's greater complexity in the brain cells of rats that were raised in enriched environments than those that were raised in impoverished environments. What greater complexity allows these brain cells to do is make more connections with other parts of the brain and therefore communicate better. A similar study was performed to examine the effect of environment and recovery from injury using enriched versus standard environment. In this experiment, they purposefully created a stroke in rats, all of them, and then gave them a tissue transplant for their brains. And then, following the tissue transplant, they provided them either with an enriched environment or the standard environment. And then they observed their response to recovery. So they tested them after a certain amount of time for function gain. So they checked their balance and agility. And almost all the rats that were raised in enriched environments, shown here in blue, demonstrated a gain of function compared to those animals that were raised in standard environments. So this indicates that enriched environments can influence not only development of the brain, but also recovery of the brain from injury. So I'm going to tackle some research methods here. I'm sure you've had this in other courses as well, but just to make sure everybody's on the same playing field, typically when you're studying something, you're conducting research, you make an observation that is of interest to you. You ask a question, and based on that question, develop a hypothesis, which is an educated guess based on known factors. Based on this hypothesis, you design an experiment to answer your question. The experiment is going to contain independent and dependent variables. So independent variables tend to be those manipulations that experimenters perform within an experiment. So, for example, if you're interested in the effect of maternal influence on brain development, you would manipulate some rats having good maternal input and other rats that maybe were neglected. These would be your independent variables. In that same experiment, the dependent variables would be factors that you use to measure brain development. So dependent variables tend to be those variables that experimenters measure, and their effect often is dependent, this is why they're called dependent variables, on what the independent variables were. You then collect the data, and based on that data, you either refute or accept the hypothesis. And then if you have to refute the hypothesis, you might revisit that hypothesis and restructure it or come up with a new hypothesis and a new experiment. So research is actually very cyclical in nature. So researchers kind of use this framework to study the effect of natural and artificial enrichment in the environment. In fact, it was your textbook's author that conducted this study. She was curious, the students in her lab were curious about the role of artificial versus natural enrichment, and what she did was she provided an environment in the lab cages that were things that a rat might encounter in the wild, and she also had a condition that was enriched with novel objects that a rat obviously wouldn't encounter in the wild. And so she delineated or distinguished these two variables, these independent variables, as natural versus artificial. She also had rats in standard cages. And then she put them through a stress test known as the forced swim test that you can see here on the bottom right. They kind of put rats in these, in a cage or a tank of water and have them swim without giving them the opportunity to escape. And at some point they just kind of stop and give up and continue to float. But this is very stressful for the rat. And so what they did in this experiment was they measured the effects of this forced swim test on the levels of a resilience hormone known as DHEA in relationship to, or the ratio of this hormone compared to a stress hormone known as corticosterone, which is a stress hormone. And so what they found was that those animals that were kind of given the opportunity to be in naturally enriched environments had more DHEA in their system, the resilience hormone, compared to corticosterone, the stress hormone. This is in comparison to the other two conditions that the animals were raised in. Furthermore, the ones that were raised in the natural enriched environment had more cells in the part of the brain associated with memory and emotional processing compared to the other two groups. So the take-home message of all of this is that an animal's environmental context can have powerful impacts on many aspects of the animal's brain and behavioral systems. Another experiment kind of demonstrating the same thing, but in this case it's a bit more powerful and possibly even applicable to the human condition, was that in the 1980s, a researcher was curious to determine if environmental enrichment can impact drug choices or drug addiction at all. So in the 1980s, there was tons of research that demonstrated that rats housed in standard lab cages would be willing to press a bar to obtain drugs like morphine and cocaine. These animals seem to have been predisposed to consuming these mind-altering drugs, just like humans were. And what's more interesting is that rats would often choose these drugs over things like water and food that they needed to survive. But a researcher, Bruce Alexander, wondered if the environment that the rats were living in was contributing to their choices. Perhaps their environment, the standard lab cage, was so dull and boring that these drugs that they were giving the opportunity to choose to take were much more exciting. So what he did was he created a rat park, which you can see here, on the right. This rat park, the animals were given an opportunity to live in, and then after a few weeks of living in this rat park, when he gave them a choice between morphine and water, these rats from the rat park almost always chose water. What's even more amazing is that some of these rats that were in the rat park were those animals that had originally chosen morphine over water in a previous experiment. So this suggests that when housed in an environment with lots of competing and engaging distractions, the excitement of the drugs is not so great, right? So I'll let you consider the ways in which we might be able to apply something like this to the human condition of addiction. So you spent a lot of time talking about the external environment influencing behavior and brain. There's also tons of research demonstrating that internal conditions can influence brain and behavior. So your digestive system hosts many, many different organisms associated with both healthy and unhealthy effects. So healthy effects may include something like digestion. Unhealthy effects may include things like an upset stomach. The colon, not to skeeve anybody out, but the colon contains about 5,600 different species of microorganisms that have been identified. There are many more that haven't been. And this is not the number of organisms, but the number of species. There are millions of bacteria in your digestive system. But these bacteria, which are known as probiotics and live in our colon, which is a part of your large intestine, helps in things like digestion. When you reduce the probiota, as it's called, in your colon, you're less likely to digest your food well and are more likely to suffer from things like, an upset stomach or diarrhea. So what they found was when researchers fed probiotics to mice, these researchers were in Ireland, these mice exhibited less anxiety and lower levels of stress hormones than mice that didn't get a probiotic diet. So examples of probiotic food that you might not realize were probiotics are, dairy products, like fermented dairy products like yogurt, kombucha, that's become popular now, sourdough bread. All of these contain probiotic organisms that help the probiota of our digestive system maintain to healthy conditions. But the point of all of this is that, our internal conditions can certainly influence our brain and behavior. And there's a whole area of neuroscience or behavioral biology, behavioral neuroscience, in trying to understand the gut-brain interaction. And this is the topic exactly that they would be studying, how the probiotic environment can influence things like anxiety levels and stress hormones. So if you're interested in something like this or interested in learning more, let me know. Interestingly enough, past and future experiences can also influence brain function and behavior, and this is called the experiential context. It is a context that's provided by past or future experiences, and we'll tackle past experiences first. So maternal support can influence brain volume and development. The greater maternal support provided to human beings resulted in greater or larger hippocampus. So the hippocampus is an area of the limbic system that we'll talk about later in the semester, and it's very important in memory as well as emotional processing as well as the stress system. And what they found was when there was greater maternal support, children that were depressed had bigger hippocampi than those children that were depressed and didn't receive greater maternal support. And another example of past events influencing brain structure, juggling training, although many of you probably wouldn't find a need for this, juggling training increased brain gray matter. So gray matter are those parts of the outer areas of our brain where nerve cells reside. So what they found was that participants that underwent greater juggling training and procured greater skills had greater gray matter in response to those improved skills. And when they stopped juggling, the brain's gray matter decreased in accordance. So the take-home message of all of this is that the brain is plastic and can be influenced by external factors, but external past factors, but there are future events that can affect the brain's response as well. So anticipation of events can influence physiology and behavior. An example of this is pain perception. Pain perception can be influenced by the unknown. If you don't know if something is awful or bad, like surgery, you might anticipate greater pain, and that's exactly what they found. When they provided patients with knowledge about a procedure and the likely level of pain they were going to experience, they reported feeling less pain when they actually underwent the surgery in comparison to those patients that didn't receive this prior knowledge. Additionally, the hippocampus, which is partly involved in the stress response, was less activated in these patients as well, these patients that were provided prior knowledge of the procedure. So the take-home message of all of this is that sometimes anticipation of future events, having knowledge of what is to come, can influence our response to that particular situation. And in this case, the uncertainty of the pain procedure can in fact increase pain perception. So let's just summarize part 1 of this week's lecture. Biological psychology is the relationship between brain and behavior. The context of a situation largely determines the brain's response to a given situation, whether it's dangerous or innocuous. Evolution has shaped our behaviors to be most adaptive to our environment. Environment can shape the brain to influence behavior, and this environment can either be internal or external, as we've discussed. Furthermore, anticipated events, those that are known or unknown, can affect the brain and body's response to those events. So that's it for this lecture. Don't forget to submit the reflection for it.